Our group studies the immune response to virus infection. Viruses cause many different diseases, ranging from the common cold to AIDS and COVID-19. COVID has illustrated that viruses really have a lot of threat to cause pandemic outbreaks. It is therefore quite important to study how our immune system is able to eradicate viruses. We are interested in a series of proteins that can be described as molecular antennas that allow cells to detect that they've been invaded and infected by viruses. And we're trying to understand how these molecular antennas work and allow our immune system to get kick-started when a virus invades our bodies. Our group uses a wide variety of techniques, ranging from basic biochemical assays all the way to in vivo modeling in mice. But most of the time we really use cultured cells from human or mouse origin. We do infections in the laboratory, where we would expose these cells to viruses and then measure how they induce immune responses. For example, the production of a set of factors called interferons that are really potent mediators of antiviral immune responses, and their name interferons stems from the fact that they interfere with virus replication. One way that allows the cells in our body to recognize that they've been infected is their ability to detect unusual nucleic acids. That could be DNA or RNA. When they detect such unusual nucleic acids, they then initiate antiviral immune responses. And over the last 5 years or so, we've discovered that DNA and RNA take on an unusual conformation called Z, in which they adopt a zigzag shape, hence the name Z, can be recognized by the innate immune system, and that activates antiviral immunity. Interestingly, this process of the recognition of unusual Z nucleic acids might also play a role in other types of diseases not involving pathogens, such as inflammatory diseases. And that's something that we are quite excited about exploring in the future as well. Our understanding of the molecular pathways that detect virus infection is moving ahead fast. A lot of this work is done in cultured cells, and it's really quite important now to understand better how these different pathways work together in an infected organism in order to mount a protective response that results in clearance of the virus. So we've got a quite diverse research group with people from many different parts of the world joining us, and we're quite proud of our diverse and inclusive culture. Everyone, although working on individual projects, really works together, and people help each other, support each other in their work. We do a lot of activities to facilitate that. For example, once a year we go away for what we call a lab retreat, which is really a scientific mini-conference from the lab and a team-building effort. And there are many other activities going on, including, for example, a cake club and others like this that create a really inclusive and fun atmosphere to work here.